My name is Melissa Franklin. I'm from this building. I'm a quantum physicist. I'm just introducing. We're not going to talk about quantum mechanics.
All right, a whole new.
A whole new thing. We have a whole new thing. I just want to talk, introduce the speaker and the other speaker, the two speakers. And this is a book here that you can get your hands on and even get it signed afterwards. But I just wanted to say, it's called Is Earth Exceptional? And I fell in love with the table of contents. And then I started reading, and then I stopped to think. But I just wanted to read a few things from the table of contents. I know that seems weird, but it just is, you're going to want to buy the book. So a freak chemical accident or a cosmic imperative. That took me like 4 hours to get through, just the title, thinking about it. The origin of life, the RNA world, I got through that. The origin of life, amino acids, the road to the protocell, from astrophysics to geology to chemistry and biology. And at that point, all of a sudden, having never taken chemistry in my life, I felt like I really need to study chemistry right away. And then there's some, is this exciting?
Yes.
Life as we don't know it, the design of natural and unnatural life forms. I was a little bit scared to read that, so I read it in the morning and not at night. The hunt for intelligence, the searches, and then the epilogue, which is very exciting, an imminent breakthrough. So that's pretty good, eh? So Jack Szostak used to be here, and I used to just run into him, and it was fun. And we would talk about physics, and the terrible thing was that I don't think he's a physicist, but he seemed to know as much physics as I did. And I'm not sure he's a chemist, but he definitely knows a lot of chemistry. Anyway, he left here. It was sad, but he's at the University of Chicago, which was also nice. And not only did he leave here, he won a Nobel Prize, something like that. I don't know exactly, maybe a Nobel Prize in medicine or physiology in 2009. And then I finally understood tonight what he won it for. So those little telomeres that are on the end of chromosomes, it keeps them from, getting wrecked in your backpack. Yeah, he figured those out with some other people. That was pretty cool. So, and then, and some other stuff. And then he went full RNA, and he's trying to figure out the origins of life. And I think we're going to hear tonight whether he's still on the RNA world hypothesis or not. Just like RNA came first, and then, but then I kept thinking, did you keep thinking this? I kept thinking this. And maybe this is colored by recent events in the world. I kept thinking, a tree is alive, right? And then I thought, are trees more intelligent than humans? That took about 4 hours. Sorry, this is a weird introduction, but Jack expected a weird introduction. So Jack is going to talk for about 15 minutes, tell us about what he's doing. Dimitar Sasolov is a professor in the astrophysics department, and he's also director of the Origins of Life Center or Initiative. And then Dimitar, also interesting enough, is an astronomer who has a lab, which is is a chemistry lab, biochemistry lab, looking for the origins of life. And they're going to talk for a while, and then you're going to be able to ask questions. Okay, anyhow, I forgot to say at the very beginning, this is the Harvard bookstore, independent bookstore that we're all trying to save all the time. So please buy a book. And the Division of Science at Harvard University and the library, the Harvard libraries, put on these talks every once in a while. And I'm really glad you could come, really, really glad that you could come. And so now it's Jack.
Thank you very much. Can you all hear me?
Yeah.
Okay, great.
So thank you all for coming. It's great to see so many people come to hear about something about science, especially these days. It's great to see so many old friends and colleagues here. I also want to thank Melissa, who's been organizing this series for a long time. I thank her for inviting me. I'm not sure I would have accepted if I had actually thought that I would have to follow her introduction, which is going to be a challenge. And I also want to thank Dimitar. And so we'll be having this conversation after I give a brief introduction to the book and you know, what it's all about. So whenever I give a general talk about the origin of life, I like to begin by actually asking you guys a question. So how many of you actually think that the universe is full of life and that there's life on planets, you know, elsewhere around other stars in our galaxy and everywhere in the universe? Life is common, lots of people, right? How many of you think that life is so complicated, there's so much going on in the simplest cell, that going from inanimate molecules to something that's living is just so incredible that it's probably very rare and life could not be common in the universe. So also a lot of people, yeah. That is generally the response I get. And I find it very interesting that people have these, diametrically opposite opinions, or in some cases, very strongly held beliefs. And of course, we don't know, right? And that's why the title of the book is a question. Is Earth exceptional in the sense that maybe this is the only place in the universe that has life? Or is it just normal and unexceptional if it's just like many other planets that may be also teeming with life? So I think that's an open question. That's why I'm doing the science I'm doing. That's why Dimitar is doing the science he's doing, and lots of our colleagues are trying to figure out the answer to that question. And so So this is just kind of summary of what we're doing in my lab, but I think there's lots of good reasons for having both of those opposite opinions, right? So there's a lot of stars in our galaxy, and we know that most stars have planets now, and so if you extrapolate from the surveys, there's got to be a lot of Earth-like planets orbiting sun-like stars. So there's so many places that we know could support life. What we don't know is if life could evolve, could get started, right? Is that the other reason for thinking that life might be common is this: on any young planet, there's a lot of time available. So on the early Earth, there's a period of roughly 700 million years from when our planet cooled down enough to support life to the first incontrovertible signs of life. So somewhere in that 700 million year period, life got started. So there's lots of time, lots of places. So given that, maybe life is everywhere. On the other hand, if you look at any cell on the planet, it's filled with thousands of moving parts, thousands of really complicated, sophisticated enzymes. They're catalyzing thousands of chemical reactions that somehow all manage to go along smoothly in concert without interfering with each other and causing the whole thing to crash. And so how could such a complicated system ever get started, right? That's been the mystery for a long time. Okay. And so basically the book tries to address this question in a few different ways. And I think one of the points that my co-author Mario Livio likes to make is to come back to the Copernican principle, right? So we know our planet's not the center of the solar system's not the center of the galaxy's not the center of the universe. We're just a regular, average, old, run-of-the-mill place in the universe. nothing special. And maybe that should apply to this question of life, right? Why would Earth be different from anywhere else? So, but it's an open question. Okay, so on that note, I did want to give a lot of credit to Mario, because without him, this book wouldn't exist. He just cold called me one day and said, "Hey, Jack, do you want to write a book on the origin of life with me?" And I've been asked to write a book many times before, but it's a daunting prospect. But I knew that Mario was a good writer. He's a good popular science writer. He has great skills at getting complicated ideas across to people. So I thought, okay, if I'm ever going to do it, this is the time, right? I should do it with Mario. And it was a very good collaboration. So he recruited me to the project, but maybe he didn't realize how hard his job was going to be to actually push me to do it and to do the work and to stay on schedule, more or less, not exactly, but more or less. So he had to do a lot of work. But we worked very well together, I think, in sort of commenting on and editing each other's work. So it was really a pleasure to work with him on this book. And it allowed us to present two different aspects of the story, right? So Mario's an astronomer, and he could present all of the ideas about life out there in the universe, exoplanets, how we're looking for life, other kinds of life that might be out there. And I could focus on the task of trying to make chemistry interesting to a public audience, which, is hard. And we had to fight with the publishers about whether we'd even be allowed to have diagrams, let alone color, which we weren't allowed to have, which is, with the technology these days, seems totally bizarre. Let's see. So there were two things that I wanted to try to accomplish in writing this book. So one, the sort of baseline thing is obvious. I just wanted to communicate as best I could the state of the art, what we've learned over recent decades, what the open questions are, and so hopefully where the field is going in the future. But the other thing that I think to me is maybe more important in the sort of larger scheme of things is to try to get across why doing science is so hard and often so slow, right? And I often wonder in this field, why wasn't this figured out 30 years ago or 50 years ago? And I think to a large extent, it's because of the fact that we all come to science with our own biases and preconceptions, and we have our own pet theories of how things ought to work. And if something doesn't work the way you think it ought to work, you have to let go of your theory or your bias in order to make progress. And I think there are many examples where that has held back progress in this field and in science at large and in many other fields of human endeavor. So I tried to give examples of that. And maybe I'll just briefly give a couple of those examples. So the famous Miller-Urey experiment, right, where they sort of passed spark energy through their idea of a primitive atmosphere and magic outcome precursors of amino acids. And it was a breakthrough at the time, right? But that work and similar analysis of the chemistry and meteorite samples and sort of exemplified by the recent studies on the sample return mission from Bennu, it led, I think, to an idea that held back progress for a long time, right? So everyone focused on the fact that it's really easy to make some of the building blocks of biology, like amino acids and even the nucleobases that you find in RNA and DNA. And what people ignored for a long time was that actually these are in parts per million or parts per billion concentration, and it's not just what you need to build biology, it's thousands of millions of other compounds. and people didn't, and so it led to this idea, this is prebiotic soup, right? And then somehow magically life emerges from this complicated mixture, which is completely ludicrous if you think about it. But it took a long, long time for people, and especially my colleague John Sutherland, to focus on that and work out how to get a small number of the right compounds at high concentrations. And that's been one of the transformative events in the field. Another example is I just wanted to end by saying one thing. And I don't want to, I want the discussion to be about science, right? But I just have to say in the current environment that if you find these puzzles interesting, if you want to know the answer to is there life out there, or how did life start here, then you have to support science. And then those answers are not going to come because the universities, including your university, are under attack, and science as a whole is under attack. And even worse than that, the rule of law is under attack. So you have to get out there and we have to. Okay.
Have a seat. Thank you, Jack. Thank you, that's great. So this is supposed to be fireside chat. So I brought some matches and we are going to set the carpet on fire soon, but first a few questions. And then when you start asking questions, the fire will. That's great. So I have to say this was very good. The audience is ready to be quizzed now, but I want us to talk a little bit more about the bigger questions that we don't know the answer to. And I have to say that I didn't quite get the answer of that question from your
That's right.
10 minutes. So
Answer, right?
what is a biochemist's view of the answer? Is Earth exceptional? And why can't you give a straight answer? What do you feel about it?
Okay. I don't think, and I mean, you're an astronomer, right? You know there's like billions of planets out there. I mean, I don't think the Earth is exceptional in many regards. The question is whether it's exceptional in having life on it, right? That's what we don't know.
And you know, people like John Sutherland would say the chemistry is tough, so it could be exceptional in the sense that it's one in a trillion chance, and then you start running out of the universe.
I think it's not quite like that. Okay, so in my view, when I look at what John and many of his students and colleagues have done, is the chemistry is actually, once you understand it, pretty straightforward. We have a lot of high yielding reactions that give us the right stuff under reasonable conditions. The part that to me still seems like it might be why life could be rare is that you have to build up, for example, that reservoir of cyanide, right? That's ferrocyanide cells. And then that has to survive for long enough until it can be thermally processed in just the right way, cooked, not too much, not too little, and then it has groundwater has to leach out the reactive compounds that are made. They have to go into the right environment for the next steps to happen, right? And you have to build up the next reservoir, and then that has to survive long enough, and then the conditions they have to get moved into a different environment, or the conditions have to change till you get to the next step. And so there's a lot of steps like this, and having all of them work one after the other might be incredibly unlikely. And I think that's very hard to address.
Sure, but at the end of this project next year, do you think you will have an answer if you manage to do part of it? All of us manage to do. No, in other words, is this a question that can be answered within the foreseeable kind of lifetime of our careers? Or do you feel that it will take longer?
Okay, so the part that we're working on in my lab is once you've got the right chemicals, how do they come together to make life? And I think we'll understand that, in a few years. And maybe our geologists, geochemists, geophysicists, friends will be able to tell us, the environments where that could happen are not too rare. So that would be nice. So I think the best we can do is string together a series of steps that look like they would be efficient, but you still have to assess the whole pathway, and I'm not sure how to do that. So the real answer to that's gonna come from you guys, right? You're gonna find signs of life out there, and just one other example will be enough for us to know that it's not that hard, right? The whole thing isn't that hard if we have one other example.
So you're passing the ball. For the last 15 years, you know, Jack was one of the founding members of the Howard Institute. That's been the story. You guys should do it, and we tell you guys should do it. But a lot of progress was made. So that's great. So let's go a little bit further in that direction. So on page 25, you say that there is one perplexing issue, you say, about modern life, and that's everything depends on everything else in a circular way.
Right.
So this is what many of you know as life is self-referential. It's a fascinating system from kind of, even if you forget the chemistry, just from a general informational and other ways. So 2 questions about that. The first one is, if life is self-referential, do we have the imagination to imagine a system that is like life, but is not self-referential? In other words, does the diversity of planets and planetary environments that we have out there beyond the solar system really map on a diverse set of biochemistries, or they all essentially converge to similar self-referential systems? What's your feeling about that?
Okay, so my feeling is, if you look at modern life, right, it's incredibly self-referential in the sense that, you know, you need RNA and proteins to make DNA, DNA to code for the RNA, which then it will code for the proteins, and so everything depends on everything else. And that is one of the conceptual bottlenecks that held back progress in this field, because life didn't start as something that's self-referential, right? It started with a much simpler form of life, the RNA world, right, which can emerge sort of directly from the chemistry of the early Earth. And so at the beginning, it wasn't self-referential. That's an evolved property of modern life. And I would expect anywhere else that we find life, it will be, you know, if it's had more than a few thousand years to evolve, it will evolve to become self-referential in that way. So I would expect that to be a universal property of highly evolved life.
So in other words, just to summarize, you think this is a very important feature, not a fault. And so basically kind of a general law. Yeah. But then, okay, so that's the second part of this question is, let's think then about the galaxy. Let's say multiple occasions in which microbial life of some sort, meaning what we are describing here, emerge, maybe different biochemistries here and there, but it becomes self-referential. So if then there is an opportunity to go one step further, what now that we do, and we create a new tree of life, is that a better way to survive the cosmic environment in your thinking? Or do you think the future is into kind of machines, things which are not self-referential, that are just self-replicators, which is a different kind of paradigm? So if you encounter multiple places of life in the galaxy, would you expect it to be self-referential?
Well, you know, I mean, the reason why life here evolved to have this self-referential aspect is that, you know, if it started with RNA, right, RNA is sort of a generalist. It's not very good at storing information. DNA is much better at that. It's not very good at doing chemical reactions. Proteins are much better than that. But you can at least imagine, maybe there's some polymer that life could get started with that is actually really good at doing both things. It seems unlikely, but that would be a way to have something that maybe didn't need to evolve DNA and a different polymer for catalysis. I don't see any reason in principle why that could happen. It doesn't seem very likely, because specialization, I mean, we all do specialization, right, to do things better. So
I mean, you keep saying in the book, RNA always wins. And I know you wrote the book 2 years ago, so does it still win?
So that was with regard to a slightly different question, which is, if life was to evolve somewhere else, would it also begin with RNA? Are there simple alternatives, right, that are just as good as RNA? And so far, the answer is no. Right, everything, but we haven't looked very widely in the sort of space of chemistry. We've looked at things that are pretty closely related to RNA that could emerge from very similar chemistry, and all those closely related polymers that I talk about in here, I think, don't work as well as RNA. But that raises a really interesting philosophical question, right? If we find life, all over the universe, is it always going to have started with the same chemistry? And that seems somewhat, that puzzles me. It seems like if there's one way to get started, there must be other ways. But that I think is kind of a job for the field of chemistry over the next 20 years, if there were enough people to be interested in addressing that and enough money to do it.
I mean, people, when I give talks and people ask me about, a question along those ways, I say, well, look, I'm a practical guy. I want to finish this project first. Then once we have a system that works, then based on what we learned from it, we can start talking about alternatives. Do you feel the same way?
Well, okay, let me tell you another story. So I had a really good student in my lab about 10 years ago, and he had a friend who was a serious chemist, and they got interested in this question, right? And they said, can we just design a molecule that will work like DNA, but in an organic solvent? And they didn't tell me about this until like 6 months after they started doing it. And they had some preliminary results that were interesting. So I thought, oh, that's cool. Keep working on it. And so 5 years later, we got up to something that is like 3 units long that could copy itself in a templated way. And at that point, we gave up. Like, this is really hard. So it's, finding DNA is amazing. RNA is even more amazing, but finding something that works under really different conditions, like in a different solvent or with a totally different chemistry, that's a really hard problem.
Yet a lot of our colleagues are doing synthetic, biology, I would say, but mostly synthetic cell work. And so there is a question for you. So imagine you're almost there. That is, the whole system is there. So there is a replicase and there is some regulation and everything. And then you discover that there is one component, like some cofactor or something, which is not prebiotic at all, completely artificial. Will you put it in and then you have the system working, or are you going to say, no, we have to finish the job. It has to be all prebiotic.
For our work?
Yes.
Okay, so here's how we approach it, right? So we, as a starting point, want to create a system that looks like a cell that can grow and divide and propagate information and therefore evolve, right? That's our starting point. I'm happy to do that in any way possible, using any chemistry, using any artificial anything. I think we learn enough from these early stage models that it helps us understand, clarifies our view of what the questions and problems are, and then we can move to something that's more realistic for the early Earth, and we can go stepwise to something that is eventually much more realistic, and hopefully the end point of that will be some pathway that can actually happen on the early Earth. But I don't think we can get there all at once. There's so many questions.
As we're talking here, I realize more and more that I actually know what you're thinking. And we all agree on all this. It's very difficult for me to pretend that I'm asking questions. We've spent so much time together. Yes. But that's fascinating. So I know some of the recent work is about this transition from RNA to DNA in the prebiotic, first ever ability to do that. And I know a lot of people ask that question, and rightfully so. Okay, so you're all talking about RNA being the really preferable. RNA always wins, works in many ways. But what about when do you think DNA comes along? And I know, you're talking about we can synthesize now the DNA monomers, but at what point the cells are kind of fed up with the promiscuity of the RNA and they say, we need something stable here. DNA is the answer.
Well, I think, so again, there are sort of multiple stages of this that we have to look at, but once you have an RNA enzyme that is a polymerase, it's easy to imagine a switch of specificity that would let you, for example, copy RNA into DNA, right? Where the ribosome, right? It's an enzyme, right? It can bind substrates that it likes. I think it's relatively easy to imagine how you could start making DNA and storing information that way. But of course, the molecules to make the DNA have to be around, right? And it looks like there's some reasonable chemical pathways that do that. But then you have to say, okay, if the deoxynucleotides and the ribonucleotides are all present and mixed together at the same time, why doesn't that screw everything up, right? And that's the kind of thing we're trying to sort out.
Well, I know you're not supposed to give out the ending of the movie or of the book in this case, but the epilogue talks about an imminent breakthrough. So 2 years later, where are we with that?
I think we're very close to understanding how you can take the right set of molecules and under the right conditions, get to something that looks like a primitive cell. So that part of the overall big puzzle, I think, we'll understand that in a few years. The bigger puzzle of exactly how life started on the early Earth, that's much harder, right? But I don't know if we'll ever understand that. And then the flip side of the coin is, there are space people are getting spectra of exoplanet atmospheres. They're already looking for biosignatures. If we're really lucky, maybe the next generation of either ultra-large Earth telescopes or the next generation of space telescopes, if there's a life-bearing planet relatively nearby, maybe we'll get an answer from that.
The ball is my court again. But I think I feel the same way. I think what you're doing with the non-enzymatic self-replication and then integrating it down in a cell, the whole team, that's coming soon and it will be fantastic. It will open the door for many things.
I think that's what I hope. It opens the doors to doing new things and answering even more questions.
And I'm out of questions, so I'm opening it to you guys.
Do you think it's more likely that the astronomers will find life or some signs of life on another planet before you find a way to create it on Earth? Or vice versa? Who's going to win that race?
Yeah, I mean, I don't know. I mean, I think we kind of egg each other on, right?
And would it be helpful for you if they were to find some signature that looked like life? Would that give you any tips?
Yeah. It would be if they find signs of life on some other planet, that would tell us that it's not that hard, right? It would encourage us to sort of, okay, work out the details of the pathway. And conversely, if we find like all the steps in a pathway that look reasonable, I think that encourages the astronomers to just like work as hard as they can and they have a better chance of seeing something out there. So I think that there's a synergy to having this work from the chemists and the astronomers.
Jack, I have a question. A few years ago, there were some interesting papers. I'm not a biologist, I'm a physicist, somewhere in Science and other places, saying that life could start at these hydrothermal vents in the ocean with the sulfur chemistry and so forth. Is this kind of, boom and over, or there is something to it?
Well, so I have a rather inflammatory paragraph in the book. I think this is a great example of pathological science, right? There is absolutely no evidence that life could start in deep sea hydrothermal vents. I mean, nothing more complicated than formate, or maybe, I don't know, traces of acetate has been made. There's no UV light down there, so you can't do all the photochemical steps that you need to make the building blocks of biology. It only ever got talked about because highly evolved life is able to colonize those environments, right? That's a totally different question from whether life can start there. And so I just think it's a terrible example of a ridiculous theory that's been popularized. But it detracts from people asking the right questions. That's my humble opinion.
Question to Jack. What do you think of panspermia? That life started somewhere beyond the Earth where conditions are more favorable and inducible for the formation of life, whereas Earth condition was not, and they were transported. Well, the probability is
Right.
tiny, but
Yeah. So, and it's one of those things that it's hard to argue against in an absolute sense, but I don't see any reason for thinking that the early Earth was an inhospitable place to get life started. In fact, it seems like an almost ideal place for life to get started, and I think it did get started on the early Earth. And even if it didn't, the whole idea of panspermia just pushes back the question to some other place and time. It doesn't help you understand anything. And I don't know, maybe you can comment more on the fact that radiation damage on a trip of millions of years between stars is probably not very likely for anything.
I mean, the whole idea started when people didn't know what was the exact age of the universe, and a lot of cosmologists actually subscribe to a model where the universe is eternal in time. And then under those conditions, panspermia is quite likely because you have infinite time for it to spread. Right now, as we understand it, the age of the universe is comparable to the age of life on this planet, so it's basically very short. So that's my kind of physicist point of view.
I mean, the universe is about roughly 3 times older than the origin of life on the Earth, and it presumably took some generations of star formation
There's no time
to generate the
Yeah.
so-called metals, as you guys like to say.
Yes.
If or when we find life, what do you think it'll look like?
So yeah, there's several levels to that question. I guess the simplest thing would be if it biochemically looks like what we have here, right? Because you can easily see that how that could happen. It would be much more interesting, of course, if it was biochemically different. That would be super interesting. But of course, what we'd all probably like is if the universe is like Star Trek, right? And there's intelligent life everywhere. That would make the universe a much more interesting place, which is a reason for having this sort of wish that the universe is full of life. But yeah, I don't know how likely that is. I mean, it's a whole separate question of, the galaxy could be full of planets that are just covered in bacterial slime, right? Which, okay, for biochemists is be exciting, but for the rest of us, maybe it's not that optimal. And what are the constraints on going from like bacteria to say larger scale life and ultimately intelligent life? That's a whole set of other interesting questions.
With consideration to things like viruses and prions, do you think our definition of terrestrial and extraterrestrial life might be different?
Not really, no. I mean, viruses viruses live in cells, right? So life didn't start with viruses because you have to have cells for viruses to grow in. You know, prions have a kind of limited aspect of heritability, which I mean, it's interesting, right? Because they can form different types of aggregates that can be self-propagating, but it's not a kind of open-ended form of evolution that can give rise to greater complexity. So I also don't think it's that interesting from the point of view of evolution.
Hi, you talk about, I guess, kind of the race versus of the astronomers versus like chemists and developing those processes. Coming from like the pharma industry and seeing how much is going into AI and algorithms to understand like protein folding and all of those models and how molecules interact with proteins. So it's not just academic industry, it's huge industrial things. Where do you think computer simulations fit into that whole research endeavor?
Yeah, that's a really interesting question, right? Because things are changing so quickly. And I think, you know, we're all asking, like, what can these AI systems actually do for us? And so there's one area that I've been involved in through a collaboration, which is, you know, when we find some chemical process, I mean, if it gives us the right product under good conditions, that's fine. Okay, we could stop there, but we don't. We want to understand how it works exactly, right? And doing quantum mechanical calculations on chemical transformations in large systems it's impossible above a certain size. And so I think that's one area where a lot of people think that AI is going to be very helpful. And of course, quantum computing could be even put those together, it could be like really amazing. That might be longer off. I don't know. It's moving fast. It's hard to say. But what I don't think is going to happen is that, you know, we just ask chat GPT, okay, tell me how life got started. It's not going to know any more than we know.
So it'll say thermal vents.
It might say that because it's trained on all
Right.
the stuff that's out there, right? So that's why you're pretty.
Oh, actually do the experiment.
You think life will have a different scale if it's on a different planet or different climate, and by scale I mean in terms of space and time.
Like how large organisms?
Yeah, how large it will be or how long things will take.
Well, you know, when you get to macroscopic plants and animals, then yeah, they would obviously be affected by the gravitational field at the surface of the planet. I think that's pretty well understood. I don't think things like chemical reactions or things on the scale of bacteria are going to be affected very much by that.
So I did ask ChatGPT, how did life start? And you came in fourth. So deep hydrothermal vents win.
So you're garbage in, garbage in.
Came in third on my.
Oh, you're using Grok.
That's amazing.
Follows on the question about what life might be like. There's all kinds of science fictional notions about using different elements, like
Right.
you use ammonia instead of water because it's a fluid molecule, you
Yeah.
use silica instead of carbon. And is that just utterly ridiculous that we can dismiss instantly, or do people in your field think about that kind of thing also, and are there any ideas?
Yeah, well, silicon instead of carbon is, I forget who said this, but silicon is the chemistry of rocks and carbon is the chemistry of life. I don't remember who said that, but I think that's pretty much correct. But okay, solve that, like instead of water, just like water ammonia at low temperatures, like maybe subsurface Titan. That's pretty interesting solvent, right? I mean, that's definitely worth doing experiments to try to understand that better. When the amazing images of the surface of Titan came from the Huygens lander. Is that right? The Cassini mission, right? Huygens lander. And we saw these amazing lakes of methane and ethane that people immediately, you just can't look at that without wondering, like, could there be life in that? But that stimulated some experiments, and it turns out, if you have that kind of solvent at that low temperature, not much dissolves in that. And yeah, so that looks like a dead end. Okay. But yeah, water ammonia, I think that's more interesting. So yeah, we have to explore all these options.
So I was wondering if you think you might figure out how life evolves and how it's generated, do you also figure out why it ends? Like, it might be like, I might think about life too complex, that it's humans or animals or trees or whatever. But if, like, do you think you're gonna find an answer to that too? Why it's.
No, I don't think so. You know what? In some of my earlier work, like the telomere stuff that Melissa mentioned, right? A lot of people were excited about that as maybe part of the solution to aging. And I think there's still tons of people working on that, right? And aging is complicated. But if we understood all of the things that go wrong, maybe we can extend life significantly. Yeah, so I think that's a really interesting area to work on. But you probably know there's like all these billionaires are starting institutes now to study these kinds of things because they want to live longer.
But I understood that question also in the more general sense. If you look at life as a phenomenon, the whole biosphere, and the ability of some of it to actually leave the planet and go somewhere else, then essentially it's a phenomenon that could be eternal.
Well, you always, not eternal for like a long time, right?
Exactly.
Okay, you're an astronomer, you could probably tell me the universe is gonna run down, right? Eventually, everything goes to thermal equilibrium and nothing interesting can happen. Trillions of years.
Yeah, maybe we can figure that out one day.
We have to live. Yeah, so you need energy and raw materials for life, right? So and we know our planet's gonna be uninhabitable in roughly 1 billion years. So
Or sooner.
so yeah, for different reasons.
Hi, Jack. I had a question. You brought up that we should pass this on to astronomers and hunt for signatures of life. Do you have any molecules that you think we should actually search for in spectra of exoplanets that could be indisputable evidence for life? And if so, what is that? And
Right. I think the general answer is there's not going to be any one thing that is an indisputable sign of life. So it's more complicated than that, right? The easiest thing to look for is the presence of oxygen in the atmosphere of an exoplanet. All our oxygen comes from photosynthesis, right? So it derives from life. But there are ways that you can, in theory at least, have oxygen-rich atmospheres from a non-biological source. So that one thing is not enough. So you have to understand a lot about the planet, right? It's the radiation it's getting from the star, its size, its orbit. You would like to see other gases that are in there as well, like things that are relatively easy to detect, like methane, right? So some sign that the atmosphere is out of equilibrium, but that by itself is also not enough, right? Because everything's out of equilibrium, because you get energy coming from the star. So So it's gonna take a lot of independent lines of life to give us gradually more and more confidence in the validity of some finding like that.
Hello, mine is also a practical question. I noticed the omission of another very important biopolymer, namely lipids. And I know you also study lipids. So I guess my question is pretty simple. It's just where in the timeline of life do you think like lipid compartments, so this is like cell membranes or any type of compartment, was it that is lipid based, was important? And then on a scale of 1 to 10, how important for the formation of life are compartments?
Okay, 10 on a scale of 10. 10. And the ironic thing is that of all the building blocks of life, the lipids are the least understood, right? Especially in terms of the prebiotic chemistry that gives rise to lipids. You know, so people always say, oh, we found like fatty acids on meteorites, but you know, parts per billion doesn't cut it, right? So it's the least understood part of important prebiotic chemistry. There are some, I think, good ideas, but they need to be developed. So hopefully we'll understand that chemistry. It'll become a priority, and we'll understand it maybe in a few years.
So, I have a question. Hi. Hi, that was really fun. So I spent a lot of time ruling things out. That's what I do with my day job. But can you rule anything out at all? And if so, sorry, I don't mean that may seem aggressive.
No,
But we're in the physics building, so is there anything you can say, it's definitely not that, or it's definitely not that unless it takes 3 billion years, or it's definitely, you know?
Well, I would say it's definitely not deep sea.
You write a scientific paper which you show that it's not. It seems like really hard, like if you find something, that's fantastic. But
yeah, right. No, I mean, you're right. It's hard. It's always hard to really rule things out. But we're looking for maybe the most probable solutions. So here's something funny, right? Venus, right? Okay, I would rule out life on the surface of Venus, like it's too hot for any interesting organic chemistry. But, you know, if you're up about, I think 60 miles or kilometers.
60 kilometers.
60 kilometers, pressure is about 1 bar, the temperature is about like this. But there's, you know, these swirling clouds going by, which are like 98% sulfuric acid. Okay, so that doesn't seem like a very good place for life. But then, you know, people are still thinking about it, like Sarah Seeger and others. And she got a guy in my lab, Daniel Dostovich, interested. And so Daniel started looking at, well, can you form membranes under that kind of condition? So they made a bunch of lipids and asked, can you form membranes and vesicles that look like protocells in sulfuric acid? And amazingly, the answer is up to 70% sulfuric acid. Yes.
Wow.
Like, never would have thought that would be possible until they actually did the experiment. And the paper just came out, so you can see the beautiful pictures. I still think 98% sulfuric acid is like, you know, an non-starter.
So if somehow you found out somehow that there wasn't life, any other life anywhere in the universe, how sad would you be? Because I know some people get very, very sad.
I think that would be kind of sad, but I guess I would be a little sad. But there are probably like imperialists, like people who would want to like spread our life throughout the conquer the other worlds. Might make some people happy.
So I want to thank, this has been really great, and I'm sorry for everybody who had to sit on the stairs, but I love it. We do it all the time in the department.
You don't have time.
Well, that's the funner part. So I wanted to thank Jack and Dimitar. It was a really beautiful conversation. And thank you mostly all of you for coming, and we're gonna actually sign books and you can buy books here in this room. We have a table up there and we can have signing down here. This time, we don't have any cheese cubes. Because we're in the physics building, I'm sorry. It wasn't my decision, I can tell you, that I'm always for cheese cubes. But please come down and if you have more questions, buy a book and you can ask a question while Jack.